Well, thank you very much, Martin, for the introduction. I'm just so happy to be finally here in person, to be able to meet people and to give a lecture, seeing eyes and being able to interact a little bit more. So in this fourth lecture, I'm going to talk about the influence of the microbiota and how it can affect graft outcome. And we came upon the microbiota because we started investigating environmental factors that could influence graft rejection. And see if I can get the mouse to work. And so as you all know, the main determinant of how strong an immune response is in the host against a transplanted organ are the number of genetic differences between the donor and the recipient. Another genetic component are the genetics of the host, where some people are, or everybody's on a Gaussian distribution of how strongly they can kill a cell or how much interferon gamma they can produce, how much their cells can proliferate, and so on. But we became interested a few years ago in whether environmental factors could also influence graft rejection and transplant outcome, thinking that as patients could be weaned progressively of immunosuppression, these external factors that can affect the immune system might have a more and more prominent role. And we didn't study pollutants, but we studied high-fat diet, we studied exercise, we studied infections, as Martin alluded to, and saw that infections could prevent the induction of tolerance and also break tolerance. And then we started working on the microbiota. So this is a class, a lecture on the microbiota, and so I wanted to give a little bit of background on the microbiota and have everybody be on the same page. And in my lab, and most of the field refers to the microbiota as the collection of microbes that colonize an environment. And in the human body, this is a combination of bacteria, viruses, archaea, fungi. We study mostly bacteria because they're easier to cultivate, they're easier to sequence. But this is not to say that the other components of the microbiota can't influence graft rejection. I'm sure they do. We refer to the microbiome as the genes that compose the microbiota. Some people refer to it as the biome, the environment where the microbes live, but most people call the microbiome the genes that form the microbiota. And the metagenome, these are really the collection of genes in a microbial community. And we refer to these bioses as a change from the normal homeostasis of the microbiota. Can be during disease or during changes of diet or during taking of medications and so on. So what we use to sequence the microbiota are two things, either 16S ribosomal RNA gene sequencing. This is easier, cheaper, faster, and it relies on sequencing a gene, part of a gene, the variable parts of the gene encoding the 16S ribosomal RNA. And these parts are extremely variable and can help place microbes or bacteria in a taxonomic category up to the genus level, more or less, because the sequence here is really profiles the types of microbes. Shotgun sequencing is more precise, and that relies on chopping the genome of these bacteria, sequencing the little pieces, and then assembling the genomes based on overlaps. And then both of them rely on aligning these sequences against known sequences that have been published, or calling them unknown if they haven't been cultivated yet. So the classification, the taxonomy is something that we think about. And that goes from domain, kingdom, phylum, class, order, family, genus, species. This is the mnemonic to remember that based on the first name, the first letter of each of these names. Dumb kids prefer candy over fresh green salad. And this is something that I say to myself several times, occasionally to remind myself where we are in the taxonomy. And here is the classification, for example, of a leopard, which is the domain of eukaryotes, kingdom of the animals, phylum chordate, and so on, mammals. The genus is panther, and this is the species. If you had the same thing for a dog, you would go all the way to the species of a dog, Canis canis. But then you have strains, and obviously a Chihuahua is very different from a Doberman, which is very different from, I don't know what else, a Golden Doodle, which is my dog. And so you have to think that because a bacterium has a name as a species, like E. coli, you have many different strains, and each strain is going to have different genetics and different potential then for biological functions, even if the name is identical. So this is something that's very important to remember. And this is the percentage identity that you have as you go up the classification. Okay, so facts on the microbiota. Microbial communities living on body surfaces that separate us from the environment, so the intestine, the skin, the lung, the gastrointestinal, the genitourinary tract, and anything that has surfaces, mucosa or epithelia that are in contact with the outside world can be colonized by microbiota, can be and are colonized by the microbiota. In the intestine, we have as many bacteria as we have cells, perhaps more, and certainly more genes belonging to the bacteria than the genes that belong to our own eukaryotic cells. And about 1 trillion bacteria in the colon per gram of intestinal content. In terms of intestinal bacteria, we have probably between 1,000 and 5,000 different species of bacteria, and probably more if you think about strains. And then each person is colonized at birth, and the mode of delivery, if you're born by vaginal delivery or by C-section, determines the first microbes that colonize you. And those first microbes in turn might determine your susceptibility, for example, to allergy and asthma with people born by C-section and colonized by cutaneous microbiota, being a little bit more susceptible to asthma and allergies than babies born by vaginal delivery. Then we all have a very individual microbiome that's personal to us and that's different and that can be distinguished by profiling of this microbiome with some core microbes that are mostly common over time and some core microbes that change based on diet, based on jet lag, environmental exposures. I'm sure my microbiota is different now with jet lag and Belgian food than it is in Chicago. And of course, the microbiota is essential for health. So just to show you the colonization from birth, here is a diagram of the colors representing the different phyla of bacteria in the meconium as the babies are born, and then as the babies age, with firmicutes in blue here being the most prominent in the beginning, and really it's bacteria that can digest milk because this is what babies eat in the beginning of life. And then as solid food is introduced, you have acquisition of actinobacteria, for example, in orange, and eventually within the 2 to 3 first years of life, the microbiota becomes more diverse and is pretty much set, with the larger part being firmicutes and bacteroidetes. But you can see that the bacteroidetes in abundance start after a year or so. And then if you take adults and then you sample their fecal material and sequence it over time at different time points in their adult life, you can see that there are things that change. but then there are things that are pretty constant over time. Now, this is looking, so these are all healthy volunteers, and it's comparing the composition of the microbiota in the fecal samples. So just a cross-sectional analysis, and each vertical column here is one individual patient. And so you can see that here there are healthy volunteers that have few in green firmicutes and a lot in blue bacteroidetes. But here on the right, you have some people, healthy people, who have a lot of firmicutes and fewer bacteroidetes. So you can be healthy and have a very different microbiota than your next door neighbor. But then if you do sequence the genes and look at the functional capacity of the bacteria, of the bacterial communities that colonize the intestine, it's actually pretty similar across these volunteers. So different bacterial species can have the same functional output in terms of genes and metabolites that they can produce, but still with some differences. So in terms of the essential roles of the microbiota for health, here's the textbook definition of the intestinal microbiota. And some bacteria in the intestine are essential to synthesize some metabolites that we need, like vitamin K for coagulation, essential to digest fibers that we couldn't digest otherwise, essential to detoxify some poisonous foods, essential to prevent colonization by pathogens by occupying niches and nutrients that make it more difficult for pathogens to establish because the space is taken, the nutrients are taken. And then importantly, they play a role in shaping the immune system, not only in developing tertiary lymphoid structures in the intestine, but also the fine profiling of the types of T cells and B cells that are present in the intestine and perhaps elsewhere in the body. And they do that through molecular patterns that can interact with molecular pattern receptors expressed on epithelial cells or mucosal cells at the barriers. And this is not just true for bacteria, of course, but true for viruses, gram-positive bacteria, gram-negative bacteria, fungi. And you can see, for example, that gram-negative bacteria have endotoxin, LPS, that can activate TLR4 on epithelial cells, or flagellin that can activate TLR5. And so these signals through toll-like receptors, through Vrije-like receptors, through NOT-like receptors, or through DNA sensors for unmethylated CpG motifs, activate transcription factors in epithelial cells, in antigen-presenting cells, dendritic cells, and so on, and instruct these cells into the production of specific profiles of cytokines and chemokines that then direct the immune system one way or another. And there are specific bacteria, for example, that have been shown to drive T-cell differentiation towards one pathway or another, with, for example, families of Clostridia being able to drive Treg differentiation, or this bug, segmented filamentous bacterium, being able to drive Th17 differentiation, or things like Listeria that can drive Th1 differentiation, for instance. So a very close interaction between the microbes, these pattern recognition receptors that sense the microbes being there, the inflammatory milieu at steady state, and the development of steady state immune differentiation based on the microbes that are present. All right, so then we're getting to the microbiota in transplantation, and when we started our experiments, which are going to be in the mouse to be able to address causal relationships, we knew that the microbiota was changing after transplantation, which you can imagine because patients receive prophylactic antibiotics, they have the insult and inflammation of surgery, they have immunosuppression. All these things can influence the composition of the microbiota. And there were some correlations that had been made between specific phyla being present in the intestine of transplanted patients, sometimes in lung, BL of lung transplant recipients. So correlations between certain phyla being present and episodes of graft rejection, but it wasn't clear whether it was a consequence of rejection that drove the establishment or the blooming of certain phyla, or if certain bacteria could cause rejection. And so we wanted to ask that specifically and establish this or inquire about the causality of this relationship. Does the microbiota composition affect the kinetics of graft rejection? And so we turned to a mouse model of skin transplantation, and we transplanted male skin into female recipients that were otherwise genetically similar. And so this is because the Y chromosome encodes a number of antigens that are foreign to the female recipient. So the female will mount an immune response against the antigens encoded by the Y antigen and will induce rejection of the skin in 20-25 days or so. And then to ask whether the diversity of the microbiota influence the kinetics of graft rejection in the absence of immunosuppression, we either treated normal mice that are colonized with a diverse microbiota, treated them with broad-spectrum antibiotics, and here is the list of the antibiotics that we used, every day by gavage directly into the stomach until the day of transplantation. Then we stopped. So we reduced the diversity of the microbiota at the time of transplantation and what the immune system is sensing in terms of microbiota. Or we used germ-free mice, which are completely sterile. They're not colonized by microbiota. They're kept in isolators. And we devised a surgical technique to be able to transplant these grafts in a completely sterile fashion without introducing colonization of these animals. And then we looked at the rejection kinetics. So if we just looked at the mice that are colonized, the rejection of male skin by female recipients happens 50% at 20 days, 25 days. So this is using sterile techniques, and this is using aseptic techniques. And if we looked at the mice that are treated with antibiotics or the mice that are germ-free, here in blue and purple, you can see that either reducing the diversity of the microbiota or eliminating the microbiota completely doubles the survival time of the graft or more, between doubles and triples, where 50% now will reject in 60 days, and the same more or less in both situations. So is the prolonged graft survival here due to the fact that these mice lack microbiota? You can ask them by giving FMT, fecal microbiota transfer, into these mice. And we gave germ-free mice then this fecal material orally to reconstitute their intestine every other day for about a week until transplantation. And if we took the microbiota from normal mice colonized, this was sufficient to accelerate rejection so that all the mice rejected within 30 days or so. However, if we reconstituted the germ-free mice with microbiota that's left after antibiotic treatment, so from these mice here, that didn't accelerate rejection at all, even though the total amount of bacteria that reconstituted these germ-free mice was similar in both cases. So that says something important. It says that the microbiota can causally accelerate graft rejection, or certain microbial communities can accelerate graft rejection, but that different microbial communities can have different effects, with the microbes left after antibiotic treatment not accelerating rejection, and the microbiota from before antibiotic treatment being able to accelerate rejection. So different quality by different components of the microbiota. So that's the graft outcome. Does the microbiota affect alloimmunity, the immune response against the transplant, against the eukaryotic determinants of the transplant? To address that, we followed graft-reactive T cells that are called Marilyn T cells that recognize one of the male antigens. And we do a trick, which is to label them with a dye, and we can see if the cells proliferate, we can see the dye getting diluted as the cells proliferate. And you can see that the proliferation is much greater if we look at these T cells in graft recipients that are colonized, and it's much reduced if we follow these graft-reactive T cells in mice that have received antibiotics. So if you have more diverse microbiota, the graft-reactive T cells proliferate more, and this is due to antigen-presenting cells. If we take antigen-presenting cells from normal mice that are colonized versus from antibiotic-treated mice, the antigen-presenting cells from antibiotic-treated mice in vitro can already activate T cells less than antigen-presenting cells from normal mice. And if we do gene expression profiling or RNA-seq from antigen-presenting cells from control or antibiotic-treated mice, and this is looking at antigen-presenting cells from 3 different mice, control mice and antibiotic-treated mice, you can see that the gene expression profiling is different. And in blue is less expression of these genes that are genes in the pathways of type 1 interferon signaling and of NF-kappa B signaling. And so what we think the microbiota is doing is that it's poising the antigen-presenting cells to enable them to activate T cells a little bit better, which kind of makes sense. You have microbes that are there. They're tuning the immune system. The immune system is now more ready to respond to threats. And so this is a model that was drawn by the person, one of the people who reviewed the paper, saying that in a normal mouse that has microbes colonizing both the skin and the intestine, you have this poising of antigen-presenting cells that allows the APCs to activate T cells more, make more interferon gamma, and reject the graft more quickly. In contrast, if you take germ-free mice that lack microbes in the gut and in the skin, or if you take antibiotic-treated mice that have a lower diversity of microbes, the antigen-presenting cells are less poised, less tuned, less able to activate T cells that then proliferate less, make fewer cytokines, and the graft is rejected with more slower kinetics. Okay, so then we were giving the antibiotics orally by gavage directly into the stomach, and we were seeing that we were changing the composition of the microbiota. This is just looking by 16S sequencing at the variety of microbes, of bacteria that are in the gut, and you can see that it's different before antibiotic and after antibiotic by principal component analysis if we look at the fecal samples, the bacteria in the gut. But we were also taking swabs of the skin because we were transplanting skin grafts, and we were curious as to whether the skin microbiota was changing with our antibiotic regimen. And indeed, we saw that there was a change after antibiotics, even though the majority of the antibiotics that we give by oral gavage is not absorbed. Something happens. One of them, metronidazole, is absorbed, plus the mice are in a cage and, in contact with fecal samples, and so that probably rubs on the skin, and if the composition of the fecal sample is different, the skin composition is going to be different. And this happens to us too. We all have fecal skin contamination. And so if we change the composition of our intestinal microbiota, we are going to change the composition of our microbiota elsewhere in the body, in the lung, in the skin. But so this begs the question now of, is it the skin or is it the gut microbiota that is important for when you transplant the skin? What is the microbial compartment that is modulating the allo response? I showed you we were tracking the graft-reactive T cells, and they were proliferating less well if we were giving antibiotics. but is it because we're reducing the diversity of the gut bacteria or because we're reducing the diversity of the skin bacteria? So we thought we could ask the question of whether if we had bacteria only in the skin and we prevented bacterial colonization in the intestine completely by starting with germ-free mice, we could address whether the skin microbiota could influence graft rejection. So to develop this system, we took germ-free mice, germ-free female mice, and germ-free male donors, and we painted the donor with a skin commensal, Staph epidermidis, before transplantation. And then because the mice groomed themselves and maybe could swallow some Staph epidermidis and colonize their intestine after transplantation, to prevent that, we gave them vancomycin in the water so that as they groomed themselves and Staph epi would enter their intestine, the vancomycin would kill the bacteria. And this was a protocol developed by a collaborator of ours, Yasmin Belkaid, at the NIH. And this works well. So this is just culture broth, LB broth, that we culture with swabs of the skin. And so you can see that if we paint the skin with Staph epi and we put the cotton swab in the LB broth, it becomes cloudy, which means that there are bacteria that are growing. But if we take the fecal pellets, because the mouse is taking vancomycin in the water, the intestine is sterile. So we can ask our questions, if you have bacteria only in the skin, can that accelerate rejection? And the answer is yes, and quite dramatically so, where here is the graft survival that I've shown you before in germ-free male skin transplanted into female recipients. But if the donor now has Staph epi on the skin, and so when you put that little skin graft, it is colonized with Staph epi, now that skin graft is rejected much, much faster. So how does that work? Is that augmenting the alloimmune response, the immune response against the mammalian components of the donor? We were expecting to see more proliferation of alloreactive T cells in the draining lymph nodes, but we didn't see that. And so we thought that because the Staph epi is in the graft, maybe it's not affecting the initial activation, the priming of alloreactive T cells in the lymph nodes. Maybe it's affecting the T cells after they go back to the graft and after they start having their effector function trying to reject the graft. And so to test that, we took the skin graft draining lymph nodes 10 days after transplantation or the skin graft itself. And you can see that in the draining lymph nodes, whether the skin transplant is germ-free or colonized with Staph epi, we really don't see any difference of inflammatory cytokines being present. However, if we look at the skin graft, and this is way before the skins are rejected, they look perfect at this time point, we can see an increase in most inflammatory cytokines. So we think that there is a different threshold or a different steady state level of inflammation that's given by having a commensal on the skin graft. And indeed, if we take antigen-presenting cells from the skin graft and we use them to activate the graft-reactive T cells, we can see now that they're able to induce a little bit more proliferation and interferon gamma of these graft-reactive T cells. But this is not true if we take the APCs from the lymph nodes, only if we take them from the graft. So the model here is that if we transplant a germ-free skin versus a Staph epi colonized skin, and the Staph epi will presumably colonize the rest of the skin in this host mouse. If we look in the skin graft draining lymph node, which is where the T cells specific for the graft are going to become activated, it's very similar. There's a little bit of proliferation, cytokine production. We don't see any difference. But if we look in the skin graft, so after the T cells become activated here and they migrate into the skin graft, now we see inflammatory cytokines, and we have these APCs that can activate the alloreactive T cells more than if we have APCs from a sterile graft. So the conclusion here is that the graft-resident microbiota is enhancing alloreactivity, meaning the T-cell immune response against the mammalian cells of the host, and you'll see in a second why I'm emphasizing that it's against the mammalian determinants of the host. So the graft-resident microbiota can augment alloreactivity at the effector phase of the immune response back in the graft. But we had a remaining question. We wanted to know if, in addition to augmenting the allo response, would it be possible that the host mount an immune response against the donor commensals in addition to against the mammalian determinants? And when the transplanted organ is colonized? And could this be in part responsible for the worst outcome that people usually see when they transplant colonized organs, like the intestine or the lung, versus if they transplant more sterile organs, like the heart or the kidney? And if the host mounts an immune response against the donor commensals, is that sufficient to induce some kind of damage to the transplanted graft independently of the allo response? If we can eliminate the allo response completely and be in a syngeneic situation, could we see if there is an immune response against a donor bacteria? And could we see if that is sufficient to damage the graft? So that's what we did. We started with a syngeneic graft model with and without microbiota. And we first asked whether the host can mount an immune response to a commensal on a skin graft. And to address that, we use T cells that are specific for Staph epi. And we transplanted either a germ-free skin or a skin colonized with Staph epi. And then 4 days later, we looked at whether these Staph epi specific T cells are proliferating or not. And we can look at the dye dilution that I explained before. You can see that if the skin graft is from a germ-free mouse, the Staph epi specific T cells do not proliferate, specific T cells in the host can proliferate. So indeed, the host can mount an immune response to a commensal that's present in the graft. And then if you look at the graft itself, now you can see accumulation of Staph epi reactive T cells that have proliferated and have diluted CFSE. And this is the quantitation of these proliferation in the lymph nodes and accumulation of divided cells. They're all divided by the time they go to the graft. So is that sufficient to induce some graft damage? And it is. It's not very dramatic, but it's significant. And you can see that there's a modest damage. And the damage, we devised a score to be able to quantify the damage because the grafts are not rejected, they recover. And so what we call a 4 is a graft that has a normal size, that hasn't shrunk, that has hair, that doesn't have red dots signaling inflammation. And I'm forgetting one. Inflammation, red dots, hair, pigmentation, and graft size. So those are the 4. And so you lose 1 point if you have any of those. So you have modest damage of the graft, but the grafts recover. But we reason that in a human population, the people have been exposed to commensals, to environmental antigens throughout their lives. And so they likely have not only naive T cells specific against commensals, but they probably have memory T cells specific against commensals. And so we want to ask whether this damage would be more dramatic if the host harbored memory to Staph epi, in this case, to this commensal. So to generate memory, we infected the mice with Staph epi. 1 month before transplantation, we let the memory develop. and then we transplanted the skin that was either germ-free or colonized with Staph epi, and we looked at the graft damage. And you can see this is the damage that I just showed you before in a naive host, and this is the damage of the skin graft that you see if the mouse has memory to the commensal. So definitely the damage is much more substantial if the mouse has memory against the commensal. And the recovery of the graft, we think, is because the immune system is actually killing the Staph epi and getting rid of it. And if we follow the survival of the Staph epi in the skin graft, you can see that by a month or so, which is when the grafts recover, the Staph epi is no longer detectable in the graft. So what's the mechanism of the graft damage? You know, could it be T cells, NK cells, innate cells, B cells? What is it? We think it's in part, and the other thing I wanted to mention is that this damage does not happen only with Staph epi, but it happens with other commensals. And here is an example with Staph aureus, colonized skin graft, where we induce memory to Staph aureus through infection a month before, and then we put the Staph aureus colonized graft or a germ-free graft. And you can see that if the mouse is naive to Staph epi, there is actually more damage. Naive to Staph aureus, there is more damage than with the Staph epi colonized graft. And if the mouse has memory to Staph aureus, there's actually very profound damage in this case. So is it T-cell mediated? To address that, we used T-cell receptor alpha knockout mice that don't have T-cells. And you can see that they have less of apparent damage of the skin graft that's colonized than the mice that do have T-cells. And the same is true if we deplete T-cells with antibodies, a combination of anti-CD4 and anti-CD8. The damage is less than in mice that are not depleted of their T-cells. And then finally, this is all with syngeneic grafts and looking at damage that is very apparent but is transient. But of course, in the normal situation, the patients are going to receive an allograft and not a syngeneic graft, and they're going to be receiving immunosuppression. So would we still see an impact of this anti-commensal immune response in a situation of an allograft with immunosuppression? To ask that, we induce memory or not to Staph epi, and then all our grafts for this experiment are going to be colonized with Staph epi. The grafts now are not syngeneic. We're back to the allogeneic model of a male skin graft into a female recipient. And our immunosuppressant, just because we have this in the lab, is injections of rapamycin. I think it's every other day for 2 weeks. And so what you can see is that if the mice don't have memory, if they're naive to Staph epi, rapamycin here in green can prolong survival over the mice that are not treated with rapamycin. However, if the mice have memory to Staph epi, here in purple and orange, rapamycin has actually no effect. It cannot overcome the activating effects of the commensal bacteria. So we're thinking that if the hosts have memory against these commensals, they can resist immunosuppression in this system. So our conclusion is that when you transplant an organ that's colonized, a skin graft, an intestine, a transplant, you're transplanting not only mammalian cells that are going to have different genetic determinants between the donor and the recipient, but you're also transplanting donor commensals. And so you're going to mount an immune response to both. And if you have memory against the commensals, you're going to have these two immune responses compound. I've shown you one study that shows that the commensal in the skin also augments alloreactivity at the effector phase of the immune response, and another study that shows that the immune response against the commensal can also directly damage the transplant. And so we think that this compounded, more severe immune response may explain why the organs that are colonized, like the lung and intestine, have a shorter half-life than the organs that are more sterile. So this is all that I wanted to tell you about, and I want to acknowledge Caroline Bartman, who was the initial student. She was an undergraduate student in my lab who worked for many years and started all the work with the microbiota in my lab. She established all the techniques, all the sequencing and everything. And Kevin Lee in the lab continued her work. And now the last study that's not published that I presented to you was all done by Isabella Pirazzolo, who was another undergraduate student. So these are not even PhD students. Kevin was a PhD student, but these were students still trying to learn their bachelor's in science who started these experiments. And with that, thank you for your attention, and I'll take questions.
So we have time for some questions. Alain, you start.
You mentioned the model, the Marilyn model, in which all the T cells are naive cells. Actually, I think so. What is the effect on either the graft microbiota or the gut microbiota on memory immune response against the graft? I mean.
Yeah, so we have not tested that. So memory to the donor, right? Memory to the alloreactive determinants. And so you're asking, would the microbiota of the gut be capable of further increasing the proliferation of graft-reactive T cells if they're memory? We have actually not tested that. It's an interesting question. I don't know, because proliferation is very robust when you're memory. I don't know if you could further activate those memory T cells, but we actually have not tested that.
Because in the clinical situation, you have already memory cells, cross-reactive memory cells.
Yes.
That could be
yeah, so it's going to be even worse, right? You're going to have memory cells against the mammalian cells of the donor and memory cells against the microbiota if you transplant a colonized organ. And so no wonder that it's harder to immunosuppress those colonized organs.
I have a question. In fact, transplantation as such might have an influence on the microbiota because it's some sort of equilibrium between the post-immune.
We have tested that.
Is it detrimental or?
We have tested just the surgery. If you transplant a syngeneic graft or if you transplant an allogeneic graft, because we were comparing how the microbiota would change with different immunosuppressive drugs versus with the inflammation of transplantation and so on. And in our hands, at least, we didn't see changes that we could detect by 16S sequencing. It's totally possible that by shotgun sequencing, which you can get to the level of species, you would see some differences. But the differences are not nearly as major as the differences that you find after antibiotics or even after some immunosuppressive drugs like tacrolimus, for example.
And we are extremely aggressive with the microbiota when I see the number of colonoscopies we're doing with a complete colic preparation where the whole intestine is completely cleansed. Are there any data with these microbiota now taking? What is the effect of colonoscopy? Do you recolonize it?
You do recolonize. Yeah, so you do recolonize. It takes a while, but you do recolonize. And in fact, there are several studies now and many anecdotal reports of people who have inflammatory bowel disease or irritable bowel syndrome who have a colonoscopy and, for 2 months they have no symptoms. And then, little by little, they're recolonized by their bacteria and the symptoms restart.
Other questions? No. Okay, then I think, Marisa, thank you very much.
Thank you.